Now we move on to our next most awaited session on nutrigenetics and nutrigenomics. We have a invitee speaker, Dr. Vimal KARANI. Dr. Vimal KARANI holds a PhD in molecular genetics. He has a interdisciplinary academic background with qualifications from medical genetics, bioinformatics, molecular biology, and genetic epidemiology. His primary research interests focus investigation on gene-nutrient interaction on metabolic and cardiovascular disease-related outcomes using approaches from genetic epidemiology, statistical genetics, and molecular biology. His long-term goal is to work on personalized nutrition by conducting observational studies on human interventions. His various publications focus on impact of haptics for promoting understanding different concepts in cell biology. Sir Vimal, Dr. Vimal KARANI, it's an honor to have you with us. May I kindly now request you to present your session on nutrigenetics and nutrigenomics.
Thank you so much for the introduction. Let me share the screen with you all. Are you able to see the screen?
Yes, sir, we can. You just have to switch. Yes, sir. Done.
Is that okay?
Yes, sir.
Okay. Thank you once again for the introduction. A very good afternoon to everyone and to everyone who have joined from different parts of the world. And today I'm going to talk to you all about nutrigenetics, caloric restriction, and cardiometabolic diseases. So I'm sure you must have been hearing a lot about fasting and caloric restricted diets since morning. And well, I'm going to talk about the thing, but in a completely different perspective in relation to the genetic susceptibility and in relation to cardiometabolic diseases. So given that we have a lot of people from different backgrounds, I thought I could start by giving some basics about genetic aspects. So we all know that our body is made up of different cells, and within each cell we have the nucleus, and inside the nucleus we have the genetic material, and in humans the genetic material is the DNA. And of course the DNA is made up of 4 chemical bases, which is the A, C, T, and G, adenine, guanine, cytosine, and thymine. So our human DNA is made up of nearly 3 billion of these chemical bases, or otherwise you call them as nucleotides. A nucleotide is nothing but a chemical base, a sugar molecule, and a phosphate together, which constitutes a nucleotide. So the human DNA has got 3 billion nucleotides, and within these 3 billion nucleotides, you have nearly about 25,000 genes, which have been discovered by the Human Genome Project. So what do these genes do, and what is the function of a gene? So gene basically is nothing but a part of a DNA segment that carries the information, genetic information for producing the proteins. So every gene will code for a specific protein. And what are these proteins? These proteins could be enzymes or secretory proteins or transport proteins or membrane proteins. So there are different kinds of proteins which are present in our body. And the main function of these proteins to get involved in metabolic functioning of the body. So which means that any variation or any genetic defect in the DNA can have an impact on the protein, and the defective protein will not be available to perform the metabolic functions. And that is one of the reasons why people develop cardiometabolic diseases such as obesity or diabetes or cardiovascular disease. So what are these genetic variations? So if you take this particular individual, he has this DNA sequence, you know that we have 25,000 genes in a DNA molecule. So every gene exists as 2 copies, because the DNA, it's double-stranded, and the DNA exists in 2 copies. So this is the 1 copy, and this will be the other copy. So this is what you would see for every individual. So let's assume that this population has got 5 individuals, and you can see that 99.9% of the human DNA is identical, where 0.1% carries all the genetic variations. So what is that genetic variation? So you can see that some individuals carry T and some individuals carry A. So there's a change from the T to A. So the A is basically the genetic variation. So there's a change from the T nucleotide to the A nucleotide. And this change from one nucleotide to another nucleotide is what you call as a single nucleotide polymorphism, or in short, SNP. So the SNP is the one that determines like whether a person carries a 0 copy or 1 copy or 2 copies of the genetic variation. That is, depending on whether you carry T or A, you can classify individuals into 3 different categories. That is, if you do not carry any copies of the genetic variation, that is, you don't carry any A at all, then it means that you are a 0 copy individual. That is, you don't carry any copies of the genetic variation. If you carry 1 copy of the genetic variation, that is 1 copy of the A and 1 copy of the T, it means that you are a 1 copy individual. If you carry 2 copies of the genetic variation, that is the 2 copies of the A nucleotide, it means that you are a 2 copy individual. So in general, 2 copy individuals, they have increased genetic risk compared to 0 copy and 1 copy individuals. So this clearly shows that for a given genetic variation, you can classify individuals into 3 different categories: 0 copy, 1 copy, and 2 copies. So individuals carrying 2 copies, they have increased genetic risk. So what is this genetic risk? So if you carry 0 copy, it means that you have a low genetic risk, which means that you have a decreased risk of developing the disease. So if you carry 1 copy, it means that you have a moderate genetic risk, which means that you have a moderate risk of developing the disease. And the third one is where you carry 2 copies, which means that you have a very high genetic risk. That means you have increased risk of developing the disease. So just because you carry 2 copies, it doesn't mean that you're destined to become diseased. You can overcome this genetic risk by modifying your lifestyle, that is, consuming a healthy diet and doing more levels of physical activity, which can overcome the genetic risk of developing a chronic disease such as obesity or diabetes or a cardiovascular disease or even cancer. So this concept is applicable to all the chronic disease outcomes, and this is what I'm going to focus for the next 45 minutes of my talk on nutrigenetics in relation to caloric restriction. So when you talk about the gene-environment interactions on cardiometabolic diseases, there are 2 important factors. One is a lifestyle factor and the genetic susceptibility. So it requires the interaction between these 2 factors for the development of these cardiometabolic diseases. So that's why we say genes load the gun, but the environment pulls the trigger. So this metaphor clearly explains the intricate interplay between the genes and the environmental factors. So you might carry the genes right from the day 1 of your life. You might carry the 2 copies, but it is basically the environmental stimuli, your lifestyle, your diet, your physical activity level, which determines whether you're going to develop the disease or not. So with this background, let's move on to what is nutrigenetics and what is nutrigenomics. So nutrigenetics refers to the study of the impact of the DNA sequence variations on chronic disease outcomes in response to a particular diet. For example, if I'm consuming a low carbohydrate diet and I carry 2 copies of the genetic variation, so am I going to become obese or not? In order to answer this question, I need to do a nutrigenetic study, that is looking at the interaction between my genetic variation and the dietary intake on obesity. So this is what you study as part of nutrigenetic, that is the gene-diet interaction. Whereas nutrigenomics, from the term that you see, nutrigenomics, omics refers to expression, genomics refers to gene expression, and nutrigenomics refers to the impact of the nutrient on gene expression. So how the nutrient that is entering inside your body can have an impact on gene expression. So when I say gene expression, what does that mean? That is, a gene is converted to RNA and RNA is converted to protein. That process is what you call as gene expression. That is a colloquial way of saying how a gene is being converted to a particular protein. So you're studying the gene expression in response to a particular diet, and that study is nutrigenomics. Whereas nutrigenetics deals with the genetic variations, how genetic variation respond to the dietary intake. And the findings from nutrigenetics and nutrigenomics are essential to implement personalized nutrition. So personalized nutrition is where you're giving an optimum diet for an individual depending on that individual's genetic makeup. So if I carry 2 copies, what is the kind of diet I should consume? If I carry 1 copy, what is the kind of diet I should consume? So this is what you try to do as part of a personalized nutrition strategy. So nutrigenetics is nothing but the gene-diet interactions, how your genetic factors can interact with your dietary intake on various chronic disease outcomes. So the concept of nutrigenetics and nutrigenomics all started after the discovery of this obesity gene. So this gene was discovered exactly 13 years ago by 2 independent genetic studies, and both these studies discovered this obesity gene, one in the UK population, one in Spanish population. And these studies basically showed that carrying 2 copies of the genetic variation can increase the genetic risk of obesity. So people strongly believe that if they carry 2 copies, they are destined to become obese. So the genetic epidemiologist decided to test by doing more levels of physical activity or consuming a healthy diet, can that overcome the genetic risk of obesity? So they wanted to know whether adopting a healthy lifestyle can overcome that genetic risk. But what is so unique and so special about this obesity gene? This obesity gene is otherwise called as the FTO gene. FTO stands for fat mass and obesity associated gene, and the genetic variations within this gene have been shown to be strongly and consistently associated with obesity traits in various populations, different ethnic groups, in men, in women, in adults, and also in children. So having seen such a strong effect of this gene on obesity, the first question that comes to our mind is, can this strong genetic effect, can this be modified by a lifestyle factor such as a diet or physical activity? So let's have a look at what is the status of the nutrigenetics in developed countries. So I'm going to give you a few examples. So one study that was done in the US population where we looked at the fried food consumption, the genetic factors, and obesity. So we stratified these 37,000 individuals into 3 different categories. Those who consume fried foods less than 1 time per week, 1 to 3 times per week, and more than 4 times per week. So in the first study, which was done in 9,000 individuals, as you can see, the white bar, individuals consuming high amounts of fried foods over a period of time, they had significantly higher genetic risk of gaining higher BMI. A similar finding was also observed in the next 2 studies, and when we combine all the 3 studies, we still found the same effect, where we found that the unhealthy diet, the fried foods, the fast foods, can increase the genetic risk of obesity, whereas the healthy foods, consuming less amounts of fried foods, can overcome the genetic risk of obesity. So this was basically a cross-sectional study which clearly showed that consuming a healthy diet can overcome the genetic risk. Another study which I did was basically a longitudinal study where we followed up 11,000 individuals from 5 different European countries for 3.5 years, and we wanted to know whether individuals following the Mediterranean dietary pattern. The Mediterranean diet is considered to be a healthy diet, which is rich in omega-3 fatty acids, fruits, vegetables, and olive oil. So consuming such a diet, we would expect that people can overcome the genetic risk, and that's exactly what we identified. So in the first part of our study, we found that individuals carrying the 2 copies of that obesity gene variation, they gain more weight. But when we introduce a Mediterranean diet into the model, we found that individuals consuming more amounts of Mediterranean foods, they alone were able to overcome the genetic risk of gaining weight. So irrespective of the type of the study design, whether the cross-sectional study or a longitudinal study, both the studies clearly implicated the fact that healthy diet can overcome the genetic risk of obesity. So now let's have a look at the postprandial lipaemia, because the nutrigenetics of postprandial lipaemia is very crucial when talking about caloric restrictions. A postprandial condition is where, like, after you eat something, what is happening inside your body, because that is what is happening the majority of the time, because humans spend majority of the time in a postprandial state. So for those who do not have a background on postprandial lipaemia, so it's basically a state like where you have a rise in triglyceride rich lipoproteins after eating, and it is a dynamic, non-steady state condition in which humans spend the majority of the time. And you can see that the non-fasting triglyceride concentration has been shown to be a very important clinical risk factor for cardiovascular disease, and also this rise in the triglyceride level as is being controlled by both genetic and environmental factors. So if you want to determine the postprandial lipemic condition, it is very complex and challenging because there are several genetic factors and several environmental factors which play a very important role. So if you look at this based on the typical Western eating patterns, most people, they consume nearly 3 or 4 meals a day, with each containing 20 to 70 grams of fat. So we carried out a postprandial lipemic study in the UK population. So the study name is a DISRUPT study, which is a postprandial sequential meal protocol. So what we did was basically we gave the breakfast at the baseline, and then we took the blood sample and we took the blood sample and gave the breakfast, and then we followed up the individuals for every 1 hour. We collected the blood sample until 3:30 minutes, where the lunch was provided. So the breakfast was about 49 grams of fat and lunch was 29 grams of fat. And after which, like again, we followed up the individual until 4:18 minutes, after which they were discharged. So this study actually gave an idea about what is the impact of the fat intake on postprandial lipemia, that is like impact on the postprandial triacylglycerol and postprandial insulin response. So I looked at a particular gene called as the APOB gene. APOB is basically stands for the apolipoprotein B, which is a very important protein that plays a crucial role in the transport of triacylglycerol and cholesterol. So it's a very important gene that plays an important role in lipid metabolism. So in this graph, you can see that on the x-axis, you find the time after the test breakfast. On the y-axis, you find the triacylglycerol. And these 3 lines are individuals carrying 0 copy, 1 copy, and 2 copies. And as you can see from the graph, individuals carrying 2 copies, they have significantly 44% higher triacylglycerol area under the curve compared to the 0 copy and 1 copy individuals, suggesting that if you carry 2 copies and if you're consuming a diet that is high in fat-rich foods, then that can increase your genetic risk of cardiovascular diseases. Another interesting finding was in relation to the postprandial insulin response. Here again, as you can see, the 3 lines are basically 0, 1, and 2. On the y-axis, you find the postprandial insulin response. On the x-axis, you find the time after the test breakfast. And you can see that individuals carrying 2 copies, they have like 69% higher insulin area under the curve in those individuals carrying 2 copies compared to 0 and 1 copy individuals. So all these studies clearly identify the fact that consuming a healthy diet is very much required if you carry a 2 copy of genetic variation. So the nutrigenetics of postprandial lipemia is very much important. You need to understand how it works when you are spending majority of the time under postprandial condition. So in order to understand what is happening when you follow a caloric restricted diet, because that is very much essential to translate the nutrigenetic findings to a personalized nutrition or a personalized diet. Because if you need to suggest a caloric restricted diet for an individual, that you need to understand how that individual would respond in a normal way. So accordingly, you provide a caloric restricted diet taking into account of his uniqueness. The uniqueness, what I meant by uniqueness is that looking at his basal metabolic rate and various other metabolic activities in addition to the genetic makeup, which is unique for that particular individual. So now let's move on to the nutrigenetics and caloric restriction. So with this background on nutrigenetics, nutrigenomics, and the complex diseases, and also with the postprandial lipemia, now let's see what is the response, how the DNA is going to respond in relation to a caloric restricted diet. So briefly, caloric restriction is nothing but reducing the average daily caloric intake below what is habitual without affecting the nutritional intake or without depriving of any of the essential nutrients. And of course, a lot of studies have shown that caloric restriction, it has been linked to preventing aging and also extending the lifespan of individuals. And also a lot of epidemiological studies have shown that caloric restriction can reduce the body weight and the glucose levels and also the blood pressure levels. So basically, caloric restriction has got a protective effect against the development of cardiometabolic diseases. But all these studies have been carried out in animals and humans and not taking into account of their genetic makeup. But what is the situation when you take into account of their genes or the genetic makeup, which is making that person or individual very unique? So when you look at the caloric restricted diet, like one is the intermittent fasting, which is nothing but an eating pattern that cycles between periods of fasting and eating. And there are different kinds of fasting protocols. So one is a time restricted feeding, and then alternate day fasting, 5:2 eating pattern, and religious fasting. So irrespective of like whatever kind of fasting method that you follow in terms of achieving the goal of like reducing your weight or maintaining a healthy basal metabolic rate, so the outcome is something that depends on your genetic makeup. You might have seen that like some people can tolerate different levels of fasting, whereas some people are not able to tolerate, and this is what their response would be. And I've seen like so many people like who have been following fasting, like they're so comfortable and they don't complain about fasting, and they do see a very significant and a positive result. Whereas some people who are following fasting, they are not able to tolerate. They either do it like for a week and they give up. What could be the reason? So there is indeed an underlying reason which differentiate these two categories of people, and that underlying reason is nothing but your genetic makeup. So what is the science behind fasting? So of course, like there are a lot of evidences, several molecular studies and physiological studies that have been carried out, which have clearly shown the scientific evidence behind fasting, why fasting has got a positive impact on different health outcomes. So one is the caloric restricted diets have shown to decrease the insulin growth factor 1 signaling pathway, which reduces the IGF-1 levels, and that has shown to have an impact on the anti-aging and anti-cancer effects. So this is one pathway. And the other one is about the inflammatory mechanism. That is, the caloric restricted diets have shown to reduce the levels of the production of the inflammatory cytokines. So of course, it has got an impact on anti-inflammatory effects. And the third one is where it has got a role to play in adipogenesis. That is, the caloric restriction has been shown to activate PGC1 alpha. The PGC1 alpha is nothing but the peroxisome proliferated activated receptor 1 alpha coactivator. So this transcriptional coactivator works right in the beginning of a molecular pathway leading to adipogenesis. So this activation is required for normal maintenance of the adipogenesis. Adipogenesis is nothing but the adipocyte differentiation. So how the adipocytes differentiate to produce a mature adipocyte, and this regulation of the adipogenesis requires activation of PGC1 alpha. So if you look at this particular pathway of the PGC1 alpha pathway, you can see that PGC1 alpha is working right in the beginning of the pathway. So any defect in that can have an impact on the entire mechanism. So studies have shown that the caloric restricted diets seem to activate this PGC1 alpha molecule, so which has got a positive impact on the regulation of adipogenesis. And the last one is about the oxidative damage. So it has been shown to be linked to the reactive oxygen species, lowering the levels of oxidative damage to the DNA, proteins, and lipids. So again, like if you look at this particular pathway, again, this damage is likely to happen right at the beginning of the pathway, so which could be prevented by caloric restricted diets. But all these evidences have come from animal studies like mouse models and yeast in Drosophila, which is a fruit fly. So all the animal models have supported, have given a lot of evidence in relation to the fact that caloric restricted diets do have a positive impact. And of course, like in humans, most of the studies, majority of the studies have been in vitro studies. So looking at the cellular mechanisms. But what is happening inside a human is completely different. Nobody knows what is happening. So the science behind fasting, in terms of the evidence, of course, there is a strong evidence from animal studies, but from the human studies, the evidence is a very big question. But of course, having said this, I'm not sure how many people have heard about the Sirt-food diet. So this diet became quite popular last year after this British pop singer Adele Atkins, who followed the Sirt-food diet. The Sirt-food diet is basically a diet which is rich in polyphenols. So she was consuming a diet like which is rich in all these polyphenols, like consuming loads of olive oil, green tea, turmeric, walnuts, and chilies, and the various other, the long list is over there. So The Sirt-food diet is nothing but it's a diet which consists of mainly polyphenols, and these polyphenols, they stimulate a gene called as the Sirt-1 gene. The Sirt-1 gene codes for a protein called Sirt-2-Win, which plays an important role in extending your lifespan, and where the studies have shown from animal studies, right? So after following the Sirt-food diet, she became like this. So this became a very popular news, and now all the gym instructors in the UK, they are giving the same kind of a diet to all the clients. And how it works is that the caloric restricted diet, which is the Sirt-food diet, it overexpresses the Sirt-1 gene or the Sirt-2 gene. And this overexpression basically has shown to extend the lifespan, that is making a person much more younger than before. So this is how the Sirt-food diet works. But this is an evidence from social media, but not from a published paper or a study that has been done under ethical approval. So this is something that needs to be considered under caution. So we need to take into consideration whether we can do a kind of a human intervention study to check whether the Sirt-food diet will definitely have an impact on weight loss. So the science behind fasting. So why the science is, why it is quite uncertain in terms of looking at the fasting mechanisms. So first of all, animal studies do not necessarily apply to humans, because the humans, they have a very complex system. And secondly, most of the human clinical trials are being short, either few weeks or few months. And the longest trial have been like up to 2 years. Even 2 years is a very short time to see the impact of fasting or the caloric restricted diets on different health outcomes. And thirdly, most of the clinical trials have shown, have done studies in relation to adults aged below 60 years. So what's going to happen in adults like who are above 60 years? So we do not know about that. And then the human system, there are a number of organic and chemical processes that are happening inside the human body, and which needs to be taken into account when giving a caloric restricted diet. And then humans are different from each other in terms of gender, the size, the body size, the height, weight, age, the genetics, environmental factors, the lifestyle, and various other factors which makes a person very unique. And most importantly, the genetics plays a very important role. I'll show you why in a few minutes. And then the reliable evidence is harder to obtain in human studies because humans are very different. And also it is very difficult to conduct a human intervention study in large number of samples. So all these factors clearly state that the science behind fasting is uncertain when it comes to humans. But there are several people who respond very well to fasting diets and caloric restricted diets, but while some others are not able to get a positive outcome with these caloric restricted diets. So let's have a look at some of the studies. So when you look at the genes and caloric restriction, there are several genetic variations. Let me tell you all that the human DNA has got 3 billion nucleotides. In these 3 billion nucleotides, you have nearly 25,000 genes. And within these genes and within this whole human DNA, you have nearly 3 million single nucleotide polymorphisms. I'm not joking, 3 million single nucleotide polymorphism. So for each single nucleotide polymorphism, you could be either a 0 copy or 1 copy or 2 copies. So remember, if you carry 2 copies in all these 3 million single nucleotide polymorphisms, if you carry 2 copies, then imagine what is the risk of becoming obese. So if this is going to be the genetic risk, whether the caloric restricted diets, do they have a positive impact on overcoming the genetic risk of obesity? So this is a question that everybody is interested in trying to answer. So I'm going to give you some examples of the studies that have been done in the past. So one is looking at the genes that are related to energy metabolism. So there are a few studies which looked at certain genes which play an important role in energy metabolism, and they wanted to know whether targeting these genes and giving caloric restricted diets, will that overcome that genetic risk? So the first study was basically focusing on the ADRP2 gene. The first column is about the gene. The ADRB2 is basically adrenergic beta 2 receptor, which is a very important gene that codes for a protein that plays an important role in energy metabolism. So this study was done in 76 Japanese individuals, where they were put on a 3-month behavioral intervention study, which included a combination of both a dietary intervention and exercise intervention, and they observed a significant reduction in body weight after 3 months. And the second study was done in relation to the UCP2, UCP2 is basically a gene that codes for the protein, uncoupling protein 2, which plays an important role in mitochondrial respiration. So, of course, like it's related to the energy metabolism and maintaining the basal metabolic rate. And this study was done in nearly 300 Korean people, and where the individuals were put on a 1-month weight control program, and VLCD stands for very low calorie diet. So they were put on a 700 kilocalories per day diet, and of course this study also showed a significant reduction in body fat mass. And the third study, again, it looked at the ADR-V2, which was done in 78 Spanish individuals, and this again focused on a 12-week intervention study, and again they found a significant reduction in body weight. So all these three genes in relation to energy metabolism did show a positive impact of the caloric restricted diet on weight loss. Now let's have a look at the genes that are related to appetite control, and the most important gene is the leptin. So leptin gene produces a protein called leptin. So leptin is produced by the adipose tissue, which goes to the brain and which binds to the leptin receptor in the brain membrane and signals the brain to stop the production of a protein called the NPY. NPY is a hormone, a neuropeptide Y, which controls your food intake. So once the NPY signal is stopped, your food intake will go down. So which clearly shows that the leptin plays an important role in your appetite control. So this defect in the leptin gene clearly showed that individuals, 117 French Caucasians, like who are put on a 2.5-month low-caloric diet, those individuals who had 2 copies, by overcoming, by following a low-caloric diet for 2.5 months, they were able to overcome that genetic risk of gaining more weight. So a significant weight loss was observed after 2.5 months. And the second study was on the leptin receptor gene variation, where 170 Spanish individuals were put on an 8-week intervention program, and following that 8-week intervention program, they found a significant weight loss at the end of the study. Again, with this gene, they found a very significant impact. And the last one is again another low-caloric diet, a study that was done in 179 French Caucasian, and which also clearly showed that the low-caloric diet can overcome the genetic risk of having the 2 copies and can have a significant weight loss at the end of the low-caloric diet, like over a period of 6 months. So all these studies have been carried out in small number of samples, and also you can see that the duration of the caloric restricted diet is very low, like it's about either 2 months or 3 months or maximum of 6 months. So what will happen like when we look at the obesity gene? Because obesity gene is a very strong genetic factor, and in all the population all around the world, this gene has shown to cause obesity in different populations in men, women. Now let's have a look at what is the impact of this particular FTO gene. So this study was done on the FTO gene, where they looked at 385 European individuals, and these individuals were put on a 12-month intervention program, including both diet and exercise, and there was no significant weight loss. And the second study, which was done on 200 European individuals, and where they were put on a 9-month intervention, and again there was no impact on weight loss. And the last study is very interesting because the largest study, which was done in 500 individuals and 4 years of follow-up, it's a multi-center intervention study in the Finnish population, and even in this population they did not see a significant impact on weight loss, but instead individuals who are following this intervention diet, they actually gained higher BMI. So all these studies clearly highlight the point that the studies vary with the sample size, the genetic variation, and different dietary intervention programs and different ethnicities. So there is no consistent finding with respect to nutrigenetics and caloric restriction. So even though caloric restricted diets work for some people, but overall, when taking into account of your genetic susceptibility, you cannot generalize or come to a firm conclusion that the caloric restriction works for people carrying 2 copies. No, you cannot say that, because we carry 25,000 genes, and it is not, we are not in a situation to take into account of all these genetic factors when designing a caloric restricted diet. So all these studies have been carried out in developed countries. So what is the situation in lower middle income countries? So let me give you some examples of the studies that I've done in lower middle income countries. So 6 years ago, I started this genuine collaboration. Genuine stands for the Gene Nutrient Interactions. So the main objective is to implement the nutrigenetic studies in these lower middle income countries, and these are some of the countries in which have implemented the nutrigenetic studies, and these are some of the publications, and these are the funding organizations that have supported my work. So now I'm going to give you some examples of the studies that I've done in the South Asian, Southeast Asian, and South African population. So one is actually a study that was done in the Indian population, where I found an interaction between the obesity gene and fiber intake on central obesity. So in this graph on the x-axis, you find the tertiles of dietary fiber, low, medium, and high. And these three colored bars are the blue and the stripe and the red, 0, 1, and 2 copies. On the y-axis, you find the waist circumference. The overall interaction was highly significant. And I found that individuals consuming high amounts of fiber-rich foods, they were able to reduce their waist circumference by 1.62 centimeter, despite the fact they carry 2 copies of the obesity gene. So which means that people who are carrying 2 copies, we would expect that they will be gaining more waist circumference. But instead, what happened was, these people, by consuming a healthy diet, that is a high fiber diet, of fiber-rich foods like greater than 44 grams of fiber per day, by consuming a high fiber diet, they were able to reduce their waist circumference, suggesting that consuming a healthy diet can overcome the genetic risk of central obesity. So for the Indian population, it is very much important. You need to consume more amounts of fiber-rich foods. And the second example is from another South Asian population, that is the Sri Lankan population, where we found an interaction between the genes and the carbohydrate intake. So in this graph on the x-axis, you find the tertiles of carbohydrate intake, low, medium, and high consumption. And the y-axis, you find the wasted ratio. And the red bar, high genetic risk, and white bar, low genetic risk. And you can see that individuals consuming high amounts of carbohydrate-rich foods, that is consuming more than 560 grams of carbohydrate per day, those individuals consuming high amounts of carbohydrate and those carrying high genetic risk, they had significantly higher levels of wasted ratio. That is, they have an increased genetic risk of central obesity. So again, going back to the fact that the unhealthy diet can increase the genetic risk of obesity. And the third study was done in the Indonesian population, that is a Southeast Asian population. But here we found an interaction with the protein intake on waist circumference. In this graph on the x-axis, you find the tertiles of protein intake. On the y-axis, you find the waist circumference, the red bar, high genetic risk, the white bar, low genetic risk. And if you look at individuals consuming low amounts of protein-rich foods, though they alone were able to reduce the waist circumference despite the fact they carry high genetic risk. That is, if you carry high genetic risk, it means that you will have a higher waist circumference. But people having high genetic risk, those who are consuming a low protein diet, they were able to reduce their waist circumference. So this again goes back to the fact that shows the uniqueness of this population and suggests that the intake of the protein, because Indonesians, they consume a lot of meat-related food products. So the consumption of animal protein is very, very high. So reducing that consumption might be helpful to overcome the genetic risk of central obesity. And the last example is from the Ghanaian population, from the West African population. So here we found an interaction between the dietary fat intake and the obesity genes in relation to central obesity. In this graph, on the x-axis, you find the tertiles of fat intake. On the y-axis, you find the waist circumference. And the black bar is a high genetic risk, and the white bar is a low genetic risk. And in this study, we were able to show that individuals who are consuming high amounts of fat-rich food, that is consuming more than 47 grams of fat per day, that is a mean intake, crossing that mean intake. Basically, if you carry a high genetic risk, then you're likely to gain more waist circumference compared to individuals who have low genetic risk. So this clearly shows that consuming an unhealthy diet can increase the genetic risk of central obesity. But for this Ghanaian population, we found an interaction with fat intake. So the take-home message is that from all these genuine collaboration, which has provided the first evidence of gene-diet interactions in all these lower middle-income countries, we have clearly shown that this FTO gene is a very crucial gene and which seems to interact with the carbohydrate, fat, protein, and fiber intake in relation to central obesity. The reason why I gave you an example of these studies from each population is that for you to understand that it is not the same diet that is being consumed in all the population. For the Indian population, high fiber intake was important. For the Sri Lankan population, low carbohydrate intake was required. And for the Southeast Asian population, the Indonesian population, low protein diet is required. And whereas for the African population, low fat intake is required. So this clearly identifies like uniqueness of each ethnic group, and which is clearly highlighting the point that each ethnic group has got its own genetic makeup. So when designing caloric restricted diet, ethnicity needs to be taken into account, and also the genetic susceptibility needs to be taken into account, because one person can respond to the fasting in a better way, whereas the other person might not be able to respond. And one of the main reasons could be the genetic factor or genetic susceptibility, because if he carries too many of the two copies, then his resistance is very much higher in terms of responding to the calorie restricted diets. So if you're interested in knowing more about the genuine collaboration, you can go into this publication, which was published in the Proceedings of the Nutrition Society early this year in this particular journal. Or if you want me to send you a copy, I'm happy to do that as well. So what is personalized nutrition? As I said before, after the discovery of this Human Genome Project, this project identified 25,000 genes and 3 million single nucleotide polymorphism. With all this valuable information, the source of genetic information, we are now in a position to personalize an individual's diet according to that individual's genetic makeup. If I carry two copies, what is the kind of diet I should consume? So these answers could be identified using the Nutrigenetic and Nutrigenomic studies and identifying a pathway towards personalized nutrition. So the take-home message from my talk is that the genetic factor and the lifestyle factor do not work independent of each other. Rather, they work together, they interact together, contributing to the development of obesity. So just because you carry two copies, it doesn't mean that you are destined to become obese. You can still overcome that genetic risk by modifying your lifestyle, that is consuming a healthy diet and doing more levels of physical activity. And the same applies to the caloric restricted diet as well. Because if you know your genetic factors, if you know that you are 1 copy or 0 copy or 2 copy, accordingly your caloric restricted diet can also be modified in such a way that that will suit your metabolic activities that you perform on a daily basis. So when talking about caloric restriction, aging, and cardiometabolic disease, so of course there is a link between caloric restriction and aging. There is an evidence, and of course caloric restriction and cardiometabolic diseases, we do have an evidence, and of course the link between the cardiometabolic diseases and aging. People who are highly prone to cardiometabolic diseases, they have increased aging molecular mechanisms that are happening inside the body. So we do have evidence from all these areas of research, but what is missing is about the genetic studies. So when you put a genetic factor into this whole scenario, it is very much of a very big question, because we do not have a strong evidence from the human studies whether there is a link between caloric restriction and aging, or caloric restriction and cardiometabolic disease, or cardiometabolic diseases and aging. So we definitely need to identify that strong link and understand why some people respond to fasting and why some people don't respond to fasting. So on this note, I would like to thank all my genuine collaborators, and finally, thank you all so much for listening to the talk. And if you have any questions, please do drop me an email, and I'm available on Facebook and LinkedIn as Vimal Karani, and also on Instagram as Vimal Karani. And thank you once again for listening to my talk, and happy to take any questions.
Yes, we have a question.
Yeah.
It's Dr. Shashikiran who asks whether restriction of calories is good or burning the calories by exercise.
Sorry?
Which is better, burning the calories by exercising or restricting the calories?
So from Nutrigenetics point of view, burning the calories has got like much more stronger evidence than restricting your diet. So that is the evidence that is available from the genetic studies.
And,
because my answers will be everything in relation to the nutrigenetics. So don't expect me to give a positive response to you all. And I'm not a clinician, I'm a basic scientist. I do a lot of research in relation to the genetic studies.
There are two questions from Dr. Raga Deepti.
Yeah.
She asks, do we have a database for 0 copies, that means ideal genes without SNPs, for all the 25,000 genes available in different disease conditions? Do we have a database for zero copies? He means.
We don't have a database for the zero copies. I mean, I don't understand what do you mean by a database for zero copies. For example, if you give your blood sample, I can tell you whether you're zero copy, one copy, or two copies, and I don't have the rights to publish that data to others because it's quite illegal and it's a very confidential information. So one cannot create a database of the information. But for example, we have different biobanks. There is one called as the UK Biobank where we have a data on nearly 500,000 individuals, and we have information about all these individuals. And still, I do not know who that person is, but still I will know whether that person is zero copy, one copy, or two copy. So that is the information level that is available on the internet.
There's Miss Swati BS who asks, how can we apply this nutrigenetics practically? Though because we don't have access to genetic study and testing the patient.
Yeah, okay. So we do have a lot of gene testing centers that are available all over India. There are about like 7 to 8 gene testing centers that have been running like for the last 6 to 7 years. So I know for sure that these centers are very good in testing your genes, and they have different packages. That is like a panel of genes like for obesity, a panel of genes for diabetes, a panel of genes for cardiovascular disease, and a panel of genes for PCOD. So you have like different outcomes. Like accordingly, they have selected certain genes, and they genotype. Genotyping is nothing but a protocol that you use it like for detecting whether you're 0, 1, or 2 copies. So if you just give your blood sample, they test on all these panels and they identify what is the risk of developing a particular disease based on your exercise levels, your dietary pattern, based on your food frequency questionnaire, and they get an idea of like the whole risk that is being given to you. And once they know that this is your genetic risk, and accordingly they can provide you a diet. But we do not have a dietary regimen that has been developed based on your genetic makeup in relation to caloric restricted diets. That is not available, but only a generalized diet that can be given to you, a balanced diet, which is unique for you. Or if you're interested in knowing more about the gene testing centers, please do drop me an email. I can suggest you some of the centers where you can get tested.
The next question is, what are the most influential nutrients to bring out a genetic expression or gene expression?
Sorry, what are the?
Most influential nutrients to bring about a change in genetic expression or gene expressions, she means.
So, yeah, okay. So there's nothing like a beneficial nutrients because that varies from one population to another population. As I said, in Indians, I found an interaction with fiber intake. In Sri Lankans, I found an interaction with carbohydrate intake. Indonesians, it was protein. And Ghanaian population, I found it with the fat intake. And again, in the UK population, I found it with saturated fat intake. And so it varies from one ethnicity to another ethnicity, and it is very unique. And quite recently, I published a paper in relation to the vitamin D and B12 deficiencies and how the vitamin D supplementation, B12 supplementation is required to moderate the levels of your gene expression. So it varies from population to another population, and it all depends on your genetic makeup.
Yeah, there was a question. Yeah, Dr. Kalpana asked, how do we find out individual genetic nature before we devise their diet or treatment? It comes to the same.
Yeah, so it's again the same point. Like if you go to any gene testing center, what you need to do is either you give your buccal swab, your saliva sample, or your blood sample, and they'll test all the genes. So in the blood, like they just break open the cell, they break open the nucleus, you get the entire human DNA. And as I said, the human DNA has got 3 billion nucleotides, and they might be looking at some of the genes, and they look at the genes and they identify whether you carry 0, 1, and 2. And once you see the gene report, you will have a better understanding. So yeah, if you need to know more about it, like just drop me an email and I can give you a copy of the gene report that will give you an understanding of how you would get a report if you go to a gene testing center.
Thank you, Dr. Vimal KARANI, for an excellent evidence-based presentation on how our genetics and our interplay between genetics and our lifestyle factor plays a very important role in prevention or treatment of non-communicable diseases. And also stressing on the importance of personalized nutrition and a customized exercise prescription is a way forward in prevention of a non-communicable or so-called lifestyle diseases. Thank you very much. It was indeed an honor to have you with us, and we definitely look forward to learning more from you in the near future. Thank you very much.
Thank you very much. Yeah. Thank you, Dr. Praveen, for inviting me to give this talk, and thanks to Arun for coordinating this whole webinar session. Thank you, everyone.
Thank you, sir.